I hope you can hear me. Um, um, I hope almost all of you are here. Even if not, I don't think we need to wait for any long. It's almost 10 past 8. I'm supposed to start at 8. Right, so I'm not even sure of the class size. Uh, there are about 33 now already, 32 already logged in. Yes, so I'm Kanishka Ukuela and I'm a senior lecturer uh, in zoology in the Rajarat University of Sri Lanka. Um, I'm actually a zoologist specializing on the evolution of reptiles and amphibians, but I use quite a few molecular biological tools or bioinformatic tools in my line of research because my work on the evolution of reptiles and amphibians involves a lot of genetics, and hence I am supposed to use a lot of bioinformatics as well as genetic tools. Right. So in this particular lecture series, my job is to teach you guys on the sections in relation to molecular biological databases and molecular phylogenetic analysis. So those are the sections that I will be covering in this particular lecture series, where I'll be at least doing 12 hours of classes. Right. Please let me know whether you can hear me or not. And I'm also supposed to record this and then upload the on. How about now? Can you hear me now? Please respond, guys. I mean, just don't type on the chat window. Okay, seems somewhat improved. Hmm. How do you increase the microphone? Maybe try increasing the volume in your computers. Uh, could also be. How about now?
Now clear, sir.
Right. Okay. Right. All right. Yeah. Fine. How is it now? Okay. Right. Okay, so yeah, let's not take any more time on this. Right. So as I mentioned before, yeah. So my sections will be on molecular biological databases and also molecular phylogenetic analysis. Right. So we're currently, as we always say, uh, we are currently in a stage in this world where we are constantly, connected uh, electronic devices, and not only that, we are constantly, uh, you know, okay, should start recording, right?
Recording in progress.
Okay, so we are constantly surrounded by a lot of information now, right? So the humans are apparently the ones who are generating this all of this information, and then we are currently, surrounded by so much information, and this is particularly relevant for biological data because there's so many scientists now in the world and they are generating so much data, specifically in the fields like genetics and molecular biology. Uh, so there's so much information being generated, and this information is now being stored in databases, and then these databases are also globally accessible, and so anybody can, usually anybody can access these databases. So there's so much data, and some of this data has not been actually used for, they've been used for, they were generated for a particular purpose, and then they have been used for that particular purpose only. And it seems that this particular data can be used for so many other things as well, right? So the focus of this particular lesson is to get an idea about these databases, and then also how to access information or retrieve information from these databases. So our focus for today's lesson is therefore uh, biological databases, but our major focus is actually molecular biological databases because this is a course on bioinformatics, and you guys are also following a degree program with relevant to biochemistry, that emphasis on molecular biology. Right. So in this particular lesson. Uh, I suppose at the end of this lesson that you should be able to describe the different types of molecular biological databases and select from various molecular biological databases and retrieve information and interpret the information into, most importantly, you should know your unit task, which molecular biological databases you should go to retrieve information, and then how to retrieve information, and then also use that information to answer questions relevant to your your question. In this particular lesson, I will be touching on what databases are and the types of databases, and then narrowing down to biological databases, and then finally focusing on molecular biological databases, where we'll be talking about primary, secondary, and specialized molecular biological databases. And then in the second aspect of the lesson, second section of the lesson, we will talk about how to retrieve data or how to retrieve information from these databases. And the two databases that I'm mostly focusing on is on our Intrace and then also sequence retrieval system. Right. Okay. So what is a database? Right? So we are talking about molecular biological databases. So of course, databases are not only focused on molecular biology, but they can be focused on so many things. Right? It could be a database containing information, student information in the Faculty of Graduate Sciences. Um, uh, so it can be anything. What is the purpose of having a database? Anybody from the audience? What is the purpose of having a database? What is the purpose of having a database?
Having same type of data gathered in the same place for convenience.
Yes, yeah, of course, convenience is a major concern. You have the same kind of data being stored there. Yeah, that's all true. Yeah, so one important aspect is that you store information, right? Why do you store information? For what purpose? Sorry?
To retrieve the data for further purposes in
Exactly,
future.
right? Exactly, yeah. We store data so that you can, access it and retrieve the data, and then you can, either study it or, for any kind of purpose. You might have to go back to those records, or and see, and retrieve information so that you can, ask a question, or verify certain thing. Right? So the most important thing is that databases store information, and then it can be, later on retrieved for any purpose that you want to access it for. So databases consist of two major aspects. That is the hardware, the computer hardware. You are supposed to store your information, and then you also need software, right? Software so that you can access it, right? You can further store data using the software, and then also you can also access and retrieve the information using the software. So databases consist of what we call entries, right? So entries are your information, and then to make things easy, we enter these records or entries, uh, breaking down them into fields. Right? For example, there's a screenshot of one of my own databases, a very simple database, where it contains information on different types of organisms that I work with. There's numbers assigned to them, right? Dates that they were observed or collected, right? Where they were collected, right? All kinds of information. Right? So they are all entered in different fields, right? So each row is an entry, and the entry, the information with specific information for each entry is broken down and placed in separate columns, which we call fields, right? So how do we retrieve data from a database? Right? So the best thing is to right, if we have a particular idea, if you are looking for a very specific record, and if you have a particular understanding of the uniqueness of the record, you can just go and go to a specific field and specify a particular piece of information. Right? So we call that a value. Right? We specify that particular piece of information, and then we are trying to retrieve information that is relevant to that particular information or value that we are actually value that we specified during uh, the specification. Right? So this process is called making a query. Right? If you're searching for a particular entry, a specific entry, uh, by specifying a particular information which you think that is relevant for your specific record, you say that you're making a query. Right? So databases, there are ways and means of creating queries, right? By specifying a particular value with respect to a particular field, you can select the most appropriate records uh, for your purpose. Right? So, um, so in a particular case where we are interested in a whole purpose of looking at molecular biological databases, and then the fact that there's so much information right now in this world stored in these biological databases. So one of the major things is that there's, as I mentioned before, there's so much information being stored, right? And those information or data were generated for a particular purpose, but uh, the of course it might have served its purpose, right? But still, there's so much new information that can be generated. There's so much new knowledge that can be generated from this information that has already been stored in these databases. Right? So we call this knowledge discovery, right? Where this data, so much data is there, right? Uh, but they were only used for a particular purpose. But if we are, you know, if we are knowledgeable enough, if we are skilled enough, we can use that information that information to to find new connections, right? So that is what we call knowledge discovery, right? So there's already information there, and there's certain things known from that particular data, right? But we try to find more discoveries, right? So that is what we call making connections. Right? So through making connections from already available data, we can find new things about this particular new things based on this information, right? And this is what we call knowledge discovery, and that is one of the major purposes of bioinformatics. I will, of course, talk about an example next week where I will give it to you as a small task, right? How new knowledge can be generated uh, by already existing data, right? So there are different types of databases. Right? So these databases are defined based on the the storage format, where how the data is being stored, and then how it is being retrieved. Right? So the three major types of databases are the flat file format databases, relational databases, and also object-oriented databases. Let's quickly go through these types of databases. So flat file databases. So this is the most oldest form of databases that are available and is rarely being used, right? So the data is basically stored as entries or records, one after the other, in a line, and the each record or entry is delimited or separated by a delimiter, which we call you know delimiter, and it's usually a special character line such as a vertical bar or an enter key in the keyboard, which defines or sets the next record. And so, of course, as we mentioned before, each record is broken down into fields, right? So fields are usually separated by tabs or commas, right? So tab spaces or commas will define the different fields in an entry. So these records usually exist as text file in a single table. So what you can see below is an example, right? So the fields are given as order date, region, representative item, units, unit cost total, and then each record is stored one after the other, right? Specific information is given one after the other. So one of the issues of this kind of system is that it's very difficult to search, to retrieve the data. The computer has to read through the entire file because you can't. Of course, there are fields, but there's no way of searching through these fields. Right? So this is all possible for small databases, and as I mentioned before, this is the earliest form of databases that has been used. And then we have the relational databases. So relational databases, the information is stored in small small tables, right? So tables consist of columns and rows, as we all know, right? So rows represent each data entry, right? And each data entry is broken down into fields which are represented by the columns. Right? Um, these tables are quite small, right? And then the connection between so data is stored in multiple tables, right? And then the tables. So for each entry, of course, each entry is broken down into several tables, and the connection between these tables are made possible by a common feature, right, which we call an attribute. A common feature will combine all the information, all the fields stored in each of these tables to one table. Right? So, of course, one table does not exist, multiple tables exist, but um, certain attributes common to all the different tables will unite the information stored in several different tables. So this is what we call cross-referencing. Uh, so of course, in this kind of the databases, you can execute a query, right? You can execute a search, uh, right? And then they are all linked by these common attributes. Uh, so, uh, because of the ability to execute queries and retrieve information, they are better at storing information, and more than that, information can be easily and quickly retrieved because of this particular format. And the final type of databases are the object-oriented databases. So here, the information or entries are stored as objects. Right? So what is an object? Right? So an object is a particular unit that combines data and mathematical routines that acts on the data. So what does this mean? So just each piece of information is broken down into a small object, and records us. Let's say I'll explain that again. Right? So let's say there's a data entry which has information on the students that are registered for a particular course in a university. So they include the information include the province the student came from, right? A name of the student, the different courses that students have registered for, right? So each of these pieces of information are broken down into small parts called objects, right? And then they are linked. For each student, these objects are linked, right? So these objects are linked through what we call pointers. These are all based on computer algorithms. So when the data is being entered, for each field, the data is broken down into or stored as objects, but they are still linked through what we call pointers. So when we are searching the information or trying to retrieve information about a particular student, um, so it involves navigating through the objects with the help of pointers. Right? So each object will link for each student. The objects are linked, and as a result, when you search in a particular field, that information will be retrieved, and then for all the information for a particular student will be displayed. So object-oriented databases are one of the most preferred kind of databases now, and most databases have that kind of follow this particular format or even relational format, relational database format. So object-oriented databases are more flexible, and they can be structured based on hierarchical relationships, right? And as a result, programming tasks can also be simplified, uh, uh, and uh, uh, and it lacks rigorous mathematical foundation. What I mean is that certain when computer algorithms are built to search for these, if the databases are complex and it requires more rigorous mathematical algorithms to be set up during searching. But object-oriented databases are simplified because each piece of information is broken down into objects and they are linked. So the rigorous mathematical foundation is not so necessary. So there's the only risk is that sometimes the relationships between objects can be misrepresented, but then it has to be carefully stored when links are being made when links are being made during the process of storing the information. So, in modern times, current databases include both object and relational database management system. That's basically a hybrid between the last two forms of databases that I just spoke about. That is the relational databases and then object-oriented databases. Right. So that's basically it for the types of databases that are being, types of databases uh, based on the uh, type of or mode of data being stored. Right. So now let's focus on biological databases. Right? So what is a biological database? So it's the collection of biological information collected from scientific experiments, published literature, or even computational analysis. So current biological databases use all types of database structures that includes flat files, relational, and even object-oriented databases. So biological databases uh, focus on research areas such as genomics, proteomics, metabolomics, microarray or gene expression studies, phylogenetics or phylogenomic studies, ecology, and even biodiversity sciences. Right? So I guess you know what
Recording in progress.
on metabolic pathways, right? And information on all different types of metabolic pathways happening inside the yes, inside the bodies. Right. So, biological databases can be classified by the kind of data they store or collect. Right. So, one can be molecular biological databases, which focus on sequence, molecules, and then there are types of databases called functional databases. Right? So, they include information on physiology, enzyme activities, phenotypes, and even ecology. And there could be something, certain databases called taxonomic databases, where species, organisms, and their names and taxonomic ranks are indicated. And there could also be image databases, images and other media like sounds, specific information, photographs, right? uh, and also information on body sizes, body measurements. So there are so many different types of database, but our focus is therefore in this particular lesson series is that molecular biological databases because that's what for bioinformaticians, biochemists, that's the most important thing that we are supposed to have a better understanding of, right? So what are molecular biological databases? So I've shortened the word to molecular databases, but ideally it should be molecular biological databases. Right? Molecular biological databases collect data and store data related to molecular biology, genetics, and biochemistry. So all these three fields are strongly linked. Right? So Bioinformatics uh, would mostly focus on data that is generated from molecular biological studies, genetics, and biochemistry. So, what sort of data are being stored in molecular biological databases? That include data such as DNA and RNA sequences, protein structures, protein sequences, gene expression profiles, metabolic pathways, and even molecular interactions. So this is the type of information that is being stored in molecular biological databases. So molecular biological databases can be broken down into three major categories. So they're the primary databases, secondary databases, and specialized databases. Primary, secondary, and specialized databases. So let's see what these three different types of databases are when it comes to molecular biological databases. So primary databases uh, contain originally original experimentally derived biological data. So any data that is derived from experiments in biological experiments, right, that's being so that data is being stored in molecular biological databases, in primary databases, and they're usually freely accessible. Anybody can access them, and the original data coming from experiments. Right? So what do I mean by original data? So they are raw data, right? So they are the data that was initially generated in that experiment, and they are being stored in a database. So there is minimal processing. The data is there, so um, processing has been minimal, right? So data that was generated initially has been directly stored in a database. Right? There's minimal processing. That is why we say it's a raw data. Right? So when raw data is further processed, right, we don't call it raw data anymore. Right? So um, the processed data is usually or further processed data is usually not stored in primary databases. So primary databases also come in three, at least three major different forms. There are nucleotide databases, protein sequence databases, and also protein structure databases. So let's look at nucleotide databases. Uh, so nucleotide databases stores DNA and RNA sequences submitted by research globally. And so anybody from anywhere in the world can submit and store nuclear sequence data in nucleotide databases. So since they are primary databases, these sequences have undergone minimal processing. Right? So they have been submitted the way it has been generated. Right? So minimal processing has been the sequence have been subjected to minimal processing. So examples for nucleotide databases, primary databases types, nucleotide databases include NCBI GenBank, ENBL, or ENBL stand for European Molecular Biological Laboratory, and within that there is EBI or European Bioinformatics Initiative, and then European Nucleotide Archive. And so they are all EBI and ENA all comes under European Molecular Biology Laboratory. And then there's also the DNA Data Bank of Japan or DDBJ. Right? So these are the three common and widely used primary molecular biological databases where nucleotide sequences are stored. So there is this understanding or convention where if one submits DNA sequence for storage in either GenBank, right, at the same time these sequences will be available and searchable in the DNA data bank of Japan and also European Nucleotide Archive. So if you submit to DDBJ, the sequences will be available at the same time in GenBank and ENA, or if you submit them to ENA, it will be available at the same time in GenBank and DDBJ. Right? So collectively, these three institutes are called International Nucleotide Sequence Database Collaboration. INSDC stands for that. Because all the data, nucleotide data sequences are shared between the three institutes, they are called the International Nucleotide Sequence Database Collaboration. So in primary databases, you also have protein sequence databases, right? So protein sequence databases stores amino acid sequences of proteins. Right? So UniProt and protein information resources are considered as primary databases, but UniProt can also be, can come under different another categorization as well, right? Sorry. Then you also have SwissProt, right? So protein. Another example for protein sequence database is SwissProt, which comes under UniProt. I'll talk about that later on, right? And also protein information resources. So those are protein sequence databases, right? Which are all unprocessed raw protein information, protein nucleotide sequence, protein or amino acid sequence information. Then we also have protein structure databases. Right? So these are three-dimensional structures of proteins, and they can also be occasionally nucleic acids. Right? The three-dimensional structure of proteins usually determined by X-ray crystallography or nuclear magnetic resonance. Right? So three-dimensional structures of proteins are usually stored in the protein data bank, or also short they're called the PDB. Right? So these are the three major primary molecular biological databases. Right? So nucleotides are being stored, unprocessed raw nucleotide sequences are stored in the nucleotide databases. And and then protein sequence databases are stored like in institutes like Swissprot and the Protein Information Resources or PIR. And then three-man dimensional structures of proteins are stored in protein structure databases such as the PDB or Protein Data Bank. Right. So let's have a look at secondary databases. So secondary databases contain uh, computationally processed or manually curated information. Right? So this information is based on the original information from primary databases. So secondary databases, very rarely do people submit to them, but you can of course do so. But then mostly secondary databases contain information that has been obtained from primary databases, but they are further computationally processed and they're manually curated, right? So what I mean by that is that they have been, further annotated, their names have been corrected, uh, different regions of the sequences or protein sequences have been clearly defined. So secondary databases have more information, and then there's more reliability in them because they have been further purified, the further cleaned and processed, and accuracy has been increased in them, right? So, but secondary databases does not mean they are more accurate. It's basically it's suggesting that uh, the information obtained from primary databases have been further processed to provide a different type of data, but from the original primary data from the primary databases. Right? So we further processed computationally and manually are also curated, uh, further curated. So examples include the PFAM. PFAM is a database of protein families on multiple sequence alignments and hidden marker models. So these are produced based on these methods, right? But it's basically a database of protein families, PFAM. Right? And then you also have ProSite, right? A database of again protein domains, families, and functional sites. So proteins are made of different domains, right? And then proteins also belong to different protein families, and they also have different functional sites. So ProSite contains information on the protein, the domains of a particular protein, and the families these proteins belong to, and then they are also their functional sites. And then you have the RefSeq, right? RefSeq is a curated collection of non-redundant representative sequences of DNA, RNA, and protein, and it's maintained by the NCBI. Right? So, what do I mean by non-redundant? Right? So, RefSeq is supposed to be a curated collection of non-redundant representative sequences. So so as I mentioned, for primary databases, anybody can submit sequences, right? Sequences of RNA, DNA, or protein sequences, or amino acid sequences. Uh, but anybody can submit the same sequence. Like multiple people can submit the same sequences multiple times, right? So like I submit the same sequence. Somebody else can also, from the same organism, same identical sequence, which was generated in a different study, can also be submitted. So, as a result, for a particular organism, like let's say Homo sapiens, you will have multiple sequences for that particular gene itself, a particular locus itself, right? Or the entire genome, you'll have multiple information or sequences represent the same organism, right? So as a result, we say that this information is redundant, right? It's the same information, but it's being stored. And the other thing is that GenBank will never delete that information, right? So whatever you submit, it will always be there and it will be stored there. Even it's the same information given by different individuals, right? So that's never been deleted. As a result, we say that that is in redundant information, the same because it's the same information. So RefSec instead has uh, therefore curated and selected certain sequences, certain high quality sequences or high quality submissions, and maintain them as representatives for that particular gene of that particular organism. Right? So that is why we say it's a RefSec. Basically, it's a reference sequence database, right? So it's still maintained by the NCBI. So RefSec takes sequences from these different submissions that are available for a particular organism or the same gene and all, and they will select one sequence per particular organism, per particular gene, and keep them as representatives for that particular species and gene. So that's what RefSec means. Right? So rather than having multiple sequences representing multiple individuals or multiple representing the same species but coming from different individuals, instead RefSec has a curated database of sequences representing each organism and each gene. Another database is MetaPsych. Right? It's a database of metabolic pathways from curated literature. And then we have something called TreeFam. Right? Phylogenetic those represent phylogenetic trees of animal gene families. And then you also have SCOP. It includes structural classification of proteins. So proteins classified based on their structure. As I mentioned before, UniProt can also be considered as a secondary databases. A database mainly because UniProt is made from a combination of two primary databases, SwissProt and PIR. Right? So UniProt is a comprehensive protein sequence database, and it consists of functional information of proteins, right? And it's further somehow curated, and the proteins are also annotated, right? And the data is basically derived from data like GenBank, PDB, but it was created through the combination of databases from SwissProt and protein information resources. So the so and that's very characteristic of secondary databases, because secondary databases are mostly uh, based on from the information coming from primary databases. So Unifront therefore has been produced from SwissProt and PIR based mainly, both of which are actually primary databases. So apart from the information that I mentioned just before, UniProt also provides detailed sequence annotation, and that includes protein structure and function, protein family assignment, domain structure of each protein, right? And the catalytic sites, cofactor binding sites, and how proteins are being modified during post translation modification, and metabolic pathways for each of these proteins, and if they are associated with diseases, and the diseases that those proteins are associated with. Uh, right. Let's go to specialized databases. Specialized databases. So, specialized databases cater to a particular research area or an organism. Right. So so they include a very specific type of data. They don't cater to a general audience, right? For example, certain databases include the information of a particular organism, such as a uh, model organism, right? So, what is a model organism? Model organism? Can you give me examples of model organisms? examples of model organisms? So we use basically model organisms to you know study particular phenomena, right? And sometimes model organisms can be used instead of human beings. But model organisms would represent a particular group of organisms, like for example, Arabidopsis thaliana would represent plants mostly, right? So a lot of studies are done, physiological studies using Thaliana because it's simpler to use them, and then um, it would represent the entire group of plants when you are focusing studies on that. So same can be said for the fruit fly Drosophila, right? So Drosophila is a highly studied model organism. Right? Its genomes, genetics is well understood because of certain reasons, uh, certain reasons of that particular, certain qualities of that particular organism. Right? So apart from model organisms, there are certain other types of databases. For example, gene promoters, right? So gene promoters are I suppose you know what promoters are, right? So these are non-coding regions of genes uh, where gene expression is being regulated by, right? So and then also there are databases of vectors, right? Vectors are usually bacteria and viruses where we use them to transfer genes to other organisms in transgenics. Um, and then so on, right? So for model organisms include, as we all know, the mouse model, the fruit fly, the zebrafish, right? Sometimes um, rice plant, which is a very staple crop, right? Rice plant is also considered as sometimes a model organism, where it's being so, like studied a lot. But rice is not a major model organism. It's not sometimes even considered as a model organism, but because it's being used, because it's a staple diet, people use it so much. People study it so much, so therefore we have a lot of information. So rice therefore has its own website which contains uh, information on its genomes and its proteome. Right? So and then there's of course uh, different types of ribosomes, and then their databases are there. Right? HIV, which is a very important disease in human beings, so HIV sequences are also there in a separate database. Right? So zebrafish is a very commonly used vertebrate model organism for developmental studies, toxicology, genetics, and so on. So there's a separate database for zebrafish as well. And even organisms like the nematode Cenorhabditis elegans, right? So it's a widely used model organism, and uh, it's sometimes served as a multicellular eukaryote organism, and, mm, and therefore a lot of studies have been done. So there are specialized. These are all specialized database focusing on model organisms or commonly studied organisms. Right. So of course, just because there's information in these databases, it doesn't mean that they come with errors, right? So they come with errors. There are sometimes sequencing errors, and then the investigators who submitted this information did not check for them particular very well, and then you end up with these errors. And if you end up using those sequences with errors, and then the errors will also be included in our analysis as well. And the other problem is the high level of redundancy, right? So as I mentioned before, redundancy is what where the same sequences from the same organisms have been submitted by multiple authors, multiple investigators, and they are being available, right? So we have so much data focusing on the same organism, on the same region of the genome or same protein. So that's a problem, right? So that makes working a bit difficult because you have so much data from the same individual or same species in the same regions of the genome. And sometimes investigators can sometimes oh, when they label them and annotate them, they can be erroneous, right? So or sometimes annotations could be incomplete, but still they ended up being accepted to these databases, and then the errors along with these databases will of course be reflected in our data or in our analysis as well if we use those data from those databases. So so therefore it's important that we should be cautious when we are using data from molecular biological databases, and then interpret the findings carefully, and also considering the errors of these errors coming from the data coming from these databases. Right. Okay. So, uh, how to retrieve information from these molecular biological databases? Right? So, the next aspect of this lesson is to retrieve information. So, there are two popular retrieval systems for biological databases. And I'm just going to focus only on these two very common ones. They are intrace and sequence retrieval system, or SRS. Right? Um, so uh, these are commonly found sequence retrieval systems, intrace and SRS. Right? So both of these systems use what we call Boolean operators to perform complex queries. Right? Boolean operators are of course very simple. They include either and or not operators. So if you use and, the search results must contain two words that we have specified. If you use or, search for results containing either word or both. And if you use the word not, it excludes the results containing one of the words. Right? So we can uh, in in in, a, in the practical advance, there's a separate course on practicals. We can try all of these, but if you want, I can just demonstrate. Mm, Wait, just a second. Mm, mm, yeah. Anyway, I'll just give me a few seconds to get to that. Um, So let's move on to entrance, which is the most commonly used sequence retrieval system or search engine. One of the best things, or one of the things that I like about Entrace is that it has it has linked many databases, right? So therefore, if you search in entrance, at the same time, it searches for so many other. It searches in so many other places. So Interest was developed and it's currently maintained by the National Center for Biological Information or Biological Information, which is NCBI, and it comes under the National Library of Medicine of the National Institute of Health in the USA. So NCBI is part of National Library of Medicine, which comes under the purview of the National Institute of Health in the USA. So what does interest do? It allows text-based searches for a wide variety of data. Text-based searches for a wide variety of data. So includes annotated genetic sequence information. You can retrieve from that. Structural informations, citations. It also retrieves abstracts, full papers of more than 4,000 journals indexed in the database called PubMed, and they can also you can also search taxonomic data, the data on organisms or animals and plants. So So one of the good things that you don't have to search for, since yeah, at the same time it searches in so many different databases that are linked to the National Library of Medicine. You don't have to search in multiple places to look for information. Instead, in one search in interest, it will retrieve um, the relevant literature, relevant information from by searching concurrently in several different databases. So these are the databases which are concurrently searched by interest, include PubMed. It's a biomedical literature database. NCBI GenBank stores nucleotide sequences, and NCBI taxonomy database contains for nucleotide sequence, DNA, and RNA sequences from more than 100,000 species of organisms. Uh, they also search in the database called PopSet. It contains data from population genetic datasets. And Unigene, it's a transcriptome database. Genome is a whole genome sequence database, and structure includes 3D macromolecular structures, and then PubChem, it's a chemical substances because database. Right. So how do you access Intrace? So you just even if you do a Google search for Intrace, um, you can it could be lead to that. So this is the basic the home page of Intrace. Uh, and then within interest you can just go for multiple you know areas within their interest. So I could just I'll just take you to interest. Mm, so if I do a Google search, okay, I suppose you can see my web browser. So it will take you to the Google the interest search, Google interest, and it'll take you to the interest website. Right. So if you click that, um, and then again, there's option called interest. Click this, and it'll take you to interest the search website. Right. So it's part of the National Library of Medicine, right? So there are multiple options: genes, proteins, blast, genomes, clinical, and PubChem. All of those different databases are there. Right? So if you're interested in a particular gene, right? Let's say click gene, it will take you to the gene sequences retrieval uh, retrieval. And if I just type, let's say I'm interested uh, in in searching for a particular gene, right? For example, cytochrome B gene, and I've just typed the shortened name for cytochrome B gene, cyt B, and I specified it as a gene, right? So within uh, you know this uh, within square brackets, I specified that cytochrome B is a gene, and I type a space and uh, and and then I also type and, right. So that's a Boolean operator saying that I'm specifying a particular gene, right. So if you just type site B gene and then hit enter or click search, it will retrieve site B genes available for all the different organisms in CBI, right. But if I specify Homo sapiens and then within the square brackets I specify it's the organism, Homo sapiens is the organism, and then search as uh, um, it will retrieve sequences relevant to particular gene and then a particular organism, right. So sometimes you could just also specify, let's uh, say mammals, and then also specify not uh, Homo sapiens, bracket, uh, and search. So I'm now saying that retrieve sequences of mammals, excluding Homo sapiens, right? Cytochrome B gene sequences of mammals, excluding Homo sapiens. So it has retrieved 1,680 sequences, excluding mammals, uh, different. Excluding Homo sapiens, all other organisms, ratus, Norwegicus, musculus, uh, the dog and its familiaris, right? So chimpanzees, and so many different organisms. So having the and operator, I have specified for cytochrome B genes of mammals, right? But if I don't want Homo sapiens, because Homo sapiens is also a mammal, right? I have typed Homo sapiens, not Homo sapiens, and then it executed the search, and it has received everything else, all mammals, excluding Homo sapiens. Right? So this is basically how you would execute a particular query search. Uh, query search for particular sequences, right? So you can also do this for other things, like other things like proteins, right? Genomes, right? And so many. There are so many options. Right? Even you can search from books if you want, right? Genes, genomes, uh, nucleotide sequences, right? And so on. And you can select each option and then say so for nucleotides and search probably only between more than that. So it has received 175,517 sequences, nucleotide sequences of cytochrome B gene of mammals, but excluding Homo sapiens. Right. Right. Let's get back to the presentation. So this is basically how you execute a query in trace, right, using Boolean operators. Right. So let's focus on different databases that are uh, searched by interest. Right? So different databases are being searched by interest. So one of them is the NCBI GenBank. Right? Let's see, look at NCBI GenBank. Um, So GenBank is a collection of nucleic acid sequences from different organisms, and they have 112 million sequences from 300 different organisms. Right? So it's a very large database, 112 million sequences now. So coming from 300,000 different organisms. So these contains genomic data, mRNAs, cDNAs or complementary DNAs, express sequence tags or ESTs, and then also high throughput sequence, raw sequence data, and sequence polymorphisms. Right? So high throughput sequence raw data is is a kind of a new sequencing system methods, and they generate large data sets from genomes and or even uh, different samples like soil and you know, microbiomes from organisms, right? And then also sequence polymorphism, right? Set of same kind of genomic regions, and then you look at differences in the same genomic region for in nucleotide differences in the nucleotides. Um, um, on the GenBank, GenPep database is also linked. It's a protein sequence database. It's basically a conceptual translation from DNA sequences, meaning that DNA sequences or DNA sequences of protein coding genes, when they have been submitted to GenBank, uh, they have been converted using uh, uh, using the genetic code, they have been converted to their amino acid sequences. Right? So that's why we call it conceptual translations, because they are not protein coded sequences. Sorry, they are not the protein sequences, but instead DNA sequences have been translated using the genetic code. Uh, so there are multiple ways you can search for sequences in GenBank. One is the text-based PubMed search, right? You can just search for, you just do a text-based search in PubMed, but it would mostly retrieve literature related to your. You can also do something called a blast search, right? So blast search involves having your own kind of data, right? Either it could be a DNA sequence database, DNA sequence, or DNA RNA sequence, or even amino acid sequence, right? So you have the data, and then you look for similar or homologous sequences, highly evolutionary similar sequences. Again, you would just search for sequences that are similar to your evolutionary, similar to your sequences against a database, right? I'll talk about this eventually, right? And then you can also conduct a text-based search from NCBI, National Library of Medicine database, and then also NCBI taxonomy browser. So as NCBI taxonomy browser is that it's a browser that contains information of all the organisms on Earth that is being classified formally. So they will also be linked. Each organism will be linked if there are nucleotide sequences available, either RNA or DNA. There will be linked to that, right? So if there are no nucleotide references, their names will still be retrieved, but they will indicate there are no sequences relative to this in relation to this particular organism. Uh, right. So this is the homepage of NCBI GenBank, right? So even in GenBank, there are multiple options. Uh, uh, so you can conduct searches in the NCBI GenBank in the same way that we conducted queries uh, using Boolean operators. But then you can also go to uh, basically Google GenBank, right? So we take you to GenBank website. Right? So default is nucleotide database, but you can also search for different genes. Uh, uh, same kind of sequences, right? Same kind of basically the same kind of output will be a sequence will be retrieved. So what this means is that you have multiple options of retrieving the same kind of data, but still searchable within the NCBI. NCBI, right? So um, uh, other option is to search the database is go to BLAST. So if you want to, if you like, BLAST is necessary when if you have a particular nucleotide sequence, either a DNA or RNA or amino acid sequence. Right? So if you have your own information, you can search against a database in the search against a database in the GenBank. Right? So if you want to go there, just basically click the BLAST tool website, which is still under the NCBI, right? It will take you to the home page of BLAST. And I'll talk about BLAST now. Right. So BLAST stands for Basic Local Alignment Search Tool. So basically, it's again a search tool. If you have your own kind of data, nucleotide information, or mm, or or uh, or your protein coding sequence data. So BLAST has multiple options, right? You can either search for nucleotides if your sequence if you have a nucleotide sequence, either DNA or RNA, you can select the nucleotide BLAST option. If you have protein sequence, amino acid sequence, you can use the protein BLAST, right? So then there are several other options, and you are able to translate the sequence to its protein coding sequence from the gene coding sequence. And if you want to search search against a protein database, I can do that. Right? If you have a protein sequence and if you want to search against a nucleotide sequence, I can use the T-blasting option. So there are multiple options. You can do this. So how does this search happen, right? So let's say you have a sequence sequence. Let's see. Okay, I'll just demonstrate how this is being done, and then mm, uh, uh, I'll show you. Uh, uh, okay, so let's go to BLAST. Let's share the right. So you can see that. Hopefully, you can see the screen. So we are on BLAST homepage. So nucleotide sequence, right? So I pasted my sequence, which I already had with me, right? Search against basically, and also optimize for highly similar sequences. Options. Yeah, so it has retrieved. So this is the results page. It's telling you that this is the name of the organism, and then also the annotation. It's a rod visual pigment RHO RNA, right? The scientific name of the organism, and it has certain parameters: scores, total score, query cover, e-value, percentage, identity, accession length, accession number. Right? So these are parameters that will help us to pinpoint the homology or how sequence. I mean, the similarity of the sequences based on evolutionary relatedness. Okay, so I just demonstrated how basically BLAST works. If I go back to the home page again, you notice my sequence is quite long, right? Sequence, the protein, the nucleotide sequence actually. So let's see how the searches are done, right? So right. So BLAST stands for Basic Local Alignment Search Tool, right? So what basically happens is that your sequence, which we now call the query sequence, right? The sequence that I searched just now, that I had a sequence, I had no idea what it was, right? I searched against a database of sequences in the GenBank. right, to see what it is and what are the most uh, evolutionary most closely related sequences are, and that would also provide insights to us about the identity of the sequence, right? So anyway, so basically what is happening is that a sequence is searched against when against the set of sequences in the data bank. So as I told you, there are about 112 million sequences are there in GenBank, and our sequences are searched against all of these sequences in the GenBank, right? So, but just imagine, like uh, if you're going to search against this 112 million large database sequence, how long it will take, right? So if you take, let's say, if it takes, if a human being, for a human being, yeah, I don't know how long it will take. Like it will take a lifetime to search against and see the level of matching between your query sequence and the database sequences. Right? So, however, as you could see, right, it was done in few seconds. Right, it took less than 10 seconds to retrieve the results page. Right? So how is this all done, right? So as its name stands, or the shortened name stands, BLAST or Basic Local Alignment Search Tool. It's a search tool, but it aligns, right? It aligns your sequences against the database sequences, right? So let's see how this algorithm works, right? So uh, let's see, by taking very small examples. Uh, so in this uh, slide, you could see in the left hand box, you could see a sequence named as S, right? It has A, C, G, T, uh, G, A, T, A. And then there is sequence labeled D. It has GATTGA, and so on, right? So in every moment, your query sequence, which is here labeled as S, is searched against all the sequences that are available in the GenBank. Right? Let's say since we have 112 million, they'll be searched against that 112 million sequences. Um, So how is this done, right? So this is done by breaking your query sequence into small fragments, right? So these small fragments are called words. Right? So usually, our search sequences, query sequences are very large. Like so the sequence that I used before, it had around 300 nucleotides, right? 300 nucleotides. So that 300 nucleotide sequence is broken down into small overlapping sequences called words. And then usually in the GenBank, these small fragments of the larger query sequence is around 11 nucleotides long. So in this particular example, it's actually smaller. It has around, they are broken down into smaller fragments. So it's indicated as S1, S2, S3, and S4. So those are smaller fragments, smaller overlapping fragments of the sequence S. Right? Now those sequences, smaller sequence fragments, are searched against a particular sequence, against the database, one sequence at a time. So they will just go and randomly align to different places of sequences and see whether there's a match, right? And when there are matches, they give scores. So for look at fragment S1, ACGTG, right? It has matched nicely against the first sequence ACGTGA, right? And CGT, right? And CGTGA has also matched, right? Um, and G TGATA has also matched in a different place, right? And completely matched in all three cases. But in one place, TGATA has matched in another the same sequence in a different place. But there's a single mismatch, right? Let's look at the letter indicated in red. There's a single mismatch, right? Where there's a C instead of a in the query sequence, right? In the database sequence, there's a C, but in the query sequence, there's a in that particular place, right? So so matches and mismatches are scored, right? So if there are matches, they will use there's a scoring system, and then they'll score for matches will get a positive score, mismatches will get a negative score, right? And the total score will be calculated. Right? So this is the score is calculated. So if A has an A, it will be +5, right? This all similar will be a +5. If there's a dissimilarity, it will be a negative score of 4. Right? So if you have positive, like all five being correct, they'll get 25. Like it depends on the number of nucleotides. If all five sequences of your code sequence matches against your database sequence, you get 25 scores. If there was only a single mismatch, you'll get 25 minus 4, it will be 21 total score. Right? So likewise, different scores will be calculated for different fragments at different places. And the next step is once a complete match is made, new nucleotides from the sequence from the query sequence will be added, right? So added from the sites and see whether there is matches also, right? So what we call that extension. And new nucleotides are being added to the sites and see whether there are matches. They are called uh, extensions. So scoring is done in two different ways, right? So for DNA sequencers, this is very simple. But in case to match, if you had to add a gap, right? Insert a gap to your query sequence, then the scoring has changed. Right? So if there's matching after adding gaps, then the positive scores will when if there are matches, the scores will be positive for matches and mismatches -3. Right? So gaps, if the gaps had to be inserted to get a match, then uh, then unfortunately scores are slightly different and it's less, right? So if you are searching against, if you're using if your query sequence is is a protein sequence or an amino acid sequence, then the score matrix, right? The system of scoring for matches and mismatches is slightly different, and the matrix that is commonly being used is either PAM or Blossom. They are named as PAM or Blossom, right? So it's slightly more different, right? So if uh, let's say these are different. So this matrix has different names, so different amino acids being included here. So proteins are made of 20 different amino acids. So C, S, T, P, A, G are all different amino acids, right? So T for tyrosine, P for phenylalanine, A for alanine, right, and so on. Right. So if uh, C amino acid matches with C amino acid, it gets a score of 9. Right? But this one, right, S amino acid matches with the S, right, it gets plus 4, right? So this is all because uh, based on the structure of the amino acid. It's not so simple as the four nucleotides that makes the DNA or RNA, but it's slightly more different, right? So blue colors indicate more marks, more positive scores. Red colors and yellow colors indicate negative scores. So here even the mismatches have different scores, right? So not just as simple uh, as nucleotide sequences, right? So this is a scoring matrix for amino acids. This is scoring matrix for using both mismatches and matches, and even if there are gaps to be added, uh, final scores will be calculated, right? And then based on the scores, it retrieves generates a list of sequences with the best scores, right? And then another parameter called the e-value, right? The sorting parameter is usually the e-value. Uh, we've got the same kind of research page for our core research as well, which we did before. Mm, So what are these parameters, right? So let's look at the parameters. So maximum score or max score is the highest alignment score calculated from the sum of the rewards for matched nucleotides and penalties for mismatches and gaps, right? So that's what we talked about before, right? The highest alignment score calculated from the sum of rewards for matches and penalties for mismatches, and then also gaps, right? If gaps are added, matches have lowest marks and mismatches have also negative marks. And then you also have a parameter called the total score. It's the sum of alignment scores for all the segments, right? So as I told you, segments are, matching is done by small segments or fragments or words, right? So total score is calculated by adding the scores for all the different fragments, right? Uh, right. So then query cover is the percentage of query length that is included in the alignment segments. So what is meant by this is that if your query cover is 300 nucleotides, and if there's 300 or more 300 or more sequence nucleotides matching or covered in the database sequences, it means that's a 100% query cover, right? And then expect value, it's another important parameter. E value or the expect value is the number of alignments expected by chance with the calculated score or better, right? Uh, so what does this mean, right? A number of alignments expected by chance, right, with the calculated score or better. So, so what is this meaning is that expect value is basically a probability value, right? So it can range from 0 to 1, right? So if you conduct uh, uh, a search again, query search using your own sequences against the database, right? Um, and then you come across a uh, sequence, and the database retrieves the algorithm, search algorithm retrieves the sequence, and if it has if it has an value of 0, it means that there is a zero probability, right? There's a zero probability that sequence was retrieved by chance, right? So meaning that values are the ideal for your result. And so if there's something, if it's better higher than 0, it means that there's a probability above 0 that the retrieved sequence had a higher score, and and it does not actually reflect the sequence, or if that it was retrieved randomly rather than it was a actual match, right? So expect value of 0 is what we actually should go for, right? If the value is above 0, then it's an indication that there is a chance that was the sequence was retrieved by chance. Right. So then there's another parameter, that is the percentage identity, right? Percentage identity. Uh, so the percentage identity is the amount of matching between your query sequence and your database sequence, right? So let's assume that two sequence there were multiple sequences retrieved in your results page, and one sequence had a 98% percentage identity or to your query sequence, another one had 85%, right? Percentage identity. So between the two sequences, which one had the highest similarity or highest homology to your query sequence? The one that had the 98% or the 85%? I'm asking a question. Right? So percentage identity, uh, it means that uh, the level of matching between your query sequence and the database sequence. Right? So we say that percentage identity of 100 means that all the nucleotides in your query sequence matched with that particular sequence retrieved from the database. Right? So if it's 95%, uh, only 95% of the sequences, only 95% of the nucleotides in your query sequence matched against the database sequence. Let's look at this, right? This results page. Right? So uh, you could see the e-values, percentage identities, right? E-values are almost all zero, so meaning that any of the sequences that are retrieved here did not come by chance, right? Uh, so they are actually real similarity. So now so we can focus on percentage identity. Certain sequences have 100% percentage identity. Some have 98%, 85%, 96%, 96%, 95, 94, and so on. Right? So which one has the highest similarity? The one that has the highest 100% or one that has the say 96% or 98%? What do you think, guys? Which one has the highest percentage? Which one has the highest similarity? The one that has 100% identity with the database sequence, or the query sequence that had the 98% identity with the query sequence against the database sequence. what does it mean, right? So if the percentage identity is 100%, it means that all 100 sequence, if there were only 100 nucleotides in your sequence, right, all the 100 nucleotides matched, right, or basically similar to your database sequence. So if that is the case, what does it mean? Right? Means that when the percentage identity is higher, the similarity of your query sequence against the database sequence is greater. Right? Is that it? Did you get it? Okay, I hope you got it. They also have parameters such as accession length, accession length, and accession accession. So accession length. These numbers are saying that the length of this particular database sequence, it has 1389 nucleotides in length, right? And then this is the accession number. So for each accession or each sequence or nucleotide sequence in the database, we'll have a unique number, right? And that is this particular number. Right? Each entry in the sequence, each entry in the GenBank will have a very unique number, and that's it. The accession length is apparently the same. Right? They all have the same kind of length, all be submitted by the same person, right? But have different percentage identities. This one, the bottommost one has 94%, and the topmost one has 100%. Indicate that what I searched against, the database has matched 100%. All the nucleotides in my query sequence is identical to this particular sequence in the gene bank. Right. Another mm, uh, database in the GenBank, as I mentioned, is the taxonomy, and uh, it has data. It is a database of organisms, and by searching for organisms, also you can just um, search for. You can just also search for uh, different, even their sequences also. Just so if I just Google NCBI taxonomy, I'll take you to the homepage of taxonomy. Um, it's just basically, search for Homo. You don't have to even specify it's an organism. You don't have to, here you don't have to specify it as an organism. Right? You can just even go for, uh, let's see, uh, e.com. coli. Right? Escherichia coli. Right? So we can just search for if there are nucleotides available for these organisms, it will display, right? So nucleotide protein. Right? So if you click nucleotide, it will take you to all the different nucleotides available. And look at that. Right? It's almost 145 million, 14 million, right? Yeah, 14 million sequences of E. coli. Right? Because E. coli comes in so many different strains. If that's possible. Right? So you could also also search for, not search for, we take you to all the different proteins that are available. Right? 85, yeah, 85 million different types of proteins. Oh my god, yeah, all from E. coli. Yeah, so basically you can search for any organism, like say, search for a Sri Lankan endemic animal. Sri Lankan endemic animal. But it has to be, the spellings has to be correct. Works. Sure about that. Yeah. This is the scientific name of jungle fowl. Yeah, there are nucleotides as well as protein coding. There's 147 different types of the source, the whole mitochondrial genome. Uh, did the study and the so this is genome. And the length of the genome it's around uh, more than 16,000 nucleotides in length. For proteins. So this is the Sri Lankan jungle fowl or the Valikukula that we have. It's endemic to Sri Lanka. So these are the different types of proteins they have from this particular species. Yeah, so that's how you search for uh, sequence information or protein sequence information from the NCBI, NCBI taxonomy browser. Right. So 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 GenBank has various kinds of formats where the information can be downloaded. The information can be downloaded as a flat file, as in and also as FASTA format, and ASN or abstract syntax notation one format, and also XML formats. So the common types of formats are flat file and FASTA. So I will of course most talk about those. Um, so, so this is a screenshot from the flat file format, right? So I'll just take you to one of these. Go back to E. coli. Let's take go to one of the nucleotide sequences, and you can see the look what kind. um, right, so let's click one of these. Um, uh, so this is the whole genome of E. coli. Right, so it tells you various different information. So this is the accession number for that whole genome. And source is E. coli, or when anything is also E. coli, right? And it's given the classification. So that's being said that it's coming directly. There's, there's so many other information. CDS or coding sequences 5108, Ds genes 5206 genes, coding genes 4952, RNA genes 98 RNA genes, RNA right, the genes, and then if you click this, it will take you to the different regions of uh, uh, the FASTA format. Take you whole genome. Yeah, this is the whole genome of the organism. Quite long. Yeah. You can specify the length. Uh, just have let's keep this one. Right. So Lafay isolate J1, and this is telling you the region of the gene, reloop gene, partial sequence telling you half of, not the entire gene sequence is there, and it's from the mitochondrial genome. Right? And this is the accession number, unique number for that particular sequence. Right? Here again, defined as Gallus-Lafayette isolate J1, J1. So it's probably a name, particular number given to a particular individual. Right? So accession number is the same. So this point one says that it is the original version. It hasn't been uh, it hasn't been modified. Right? It hasn't been modified. It's the original format it has been submitted. Right? So if it was like point two, it means it has been revised after it was submitted. So tells you the authors of the sequences, all Sri Lankans living together and so on. This is the sequences around 659 nucleotides in length.
and then the title of the slide. Source is mitochondrial. The classification of the organism is given. So let's see another one. This is a different gene. So different types of information is given here. Even the translated protein is given. So if it's a protein coding gene, the translated amino acid sequence is also given here. So this gene is from first nucleotide to 447 nucleotides. The coding region is from 1 to 7 and 357 to 447, meaning that there are introns in this particular gene. Yeah. So, so this is the data in a flat file format. Right? So basically, the data of the entry of that particular gene sequence in the gene displayed in the gene bank in the flat file format. Right? So you can also look at the this in the FASTA format. If you click FASTA format, it will be less informative. It will have it will contain the all the descriptors, the accession number, the unique number, the species name, the gene, right? The gene name. So and there are also it's in that there are exons and then partial coding sequence, right? So and then it's given in this particular format. Right? So if you want, you can even download this. Right? You selecting send to option, so you can select download and select the format like file and select the FASTA format. It will download it exactly in this particular method. Just only these will be there when you download it. So send to file and select FASTA. It will download as is. But you can also, there are other formats as well as AXN, XML, right? But most of the time people use the FASTA format because most of the software that is being used to, examine them, and then compare them with other organisms, the same gene with other organisms, you will have to go, best is to use the FASTA format. Uh, mm. So that's this. We had to look at that file format, right? It contains quite a lot of information. If it's a published paper, uh, things tell you the journal it was published in, right? Authors and so on. First, the format is shorter, just like in this particular format, right? So the protein coding sequence, you get the number, gene name, the organism, and then the sequence. So remember, most importantly, FASTA format, it will always have this symbol indicating this is the FASTA format, right? Less than symbol before the sequence. And this is the abstract syntax notation one form. It's a rarely being used format now. Right. So SRS, right? So SRS is a sequence
Recording in progress.
blast, and then also it can also use to align sequences. You can align similar regions of the sequences together and examine in in SRS system. Right? So the standard form of Boolean operators can also be used in the SRS system, and not Boolean operators, and then also brackets can be used. So the search results will contain query sequence and sequence annotation, and then also links to literature, metabolic pathways if they are available, and other biological data, links to other biological data. So this is the home page of SARIS of the European Molecular Biology Laboratory and European Bioinformatics Initiative. Right? So you just go to the home page, you can just find the gene, protein, or a chemical, and then of course, uh, you know, basically you can just search for Google search for. So default is a search page, right? So there are options. Nucleotide, if you want, if you're searching for nucleotide sequences, uh, say pro E, so it will retrieve sequences. Yeah, spelling mistake. CBCHR. Chrome. Yeah, so it is retrieved Drosophila and so on. Right? You can of course even just go for advanced search and then build a query. See, here you don't even have to type and or not. They are of course there, right? Let's build a query. So let's say nucleotide. Mm. Mm. For this, do say Chrome, Sophie, uh, mm, Mm, fine tuning. One. So search. So it can be a and cytochrome Drosophila melanogaster and cytochrome oxidase. Oh, okay, sure. Just spelling. Identify no. See, hmm.
Recording in progress.
Hmm. So I'm not sure why what the error is here. Yeah. Let's try this. Hmm. So basically, this is the way we build queries in trace, but doesn't seem to be working in yeah. But if you just do a simple search here, it will okay. Cytochrome B is there in Homo sapiens. So you just define it like here. It just turns up. Right? If you click this, it will take you to the individual sequence information. So the entire uh, uh, interface is different from entrace. So it tells you where the gene is located in the chromosome 19 in human beings. And the length of the entire genome is 34 bytes. uh, sorry, so these are different search results. The search environment is slightly different. And it's supposed to retrieve the same kind of sequences that you get in GenBank, because sequences are being shared between the two institutes. But, um, but the entire the interface is quite different. So it's um, 'cause I'm much more used to interest and NCBI. I rarely use EBI. So the however, same thing can be achieved in different ways if you use either EBI databases or NCBI databases, NCBI GenBank databases. Right. So right. So back to the presentation. So that's basically what I want to show you about sequence retrieval system, which is part of the EMBL EBI. So, in summary, biological databases are used to store and retrieve information which will assist in discovery. Biological databases are constructed as either as flat files, relational or object-oriented methods. Based on their content, biological databases are divided into primary, secondary, and specialized databases. Most popular retrieval systems for biological database are Intrace and sequence retrieval systems. Intrace is an integrated system search engine that searches for many databases, including PubMed, GenBank, ECDA taxonomy, ECDA taxonomy database, Genome and Structure, and PubChem. The GenBank of NCBI is a collection of annotated nucleic acid sequence data from organisms, and BLAST is a search tool used to find regions of similarity between sequences by comparing the core sequence to sequences stored in NCBI GenBank. Right. Right. Any questions on biological databases? Any aspect that you didn't understand before we proceed to the next step, lesson? It's important that you get it clarified. Any section that you didn't understand, please just let me know. I'll be more than happy to explain that again. Okay, if there are no questions, I'll give you a quick break before we start the next lesson. I'll see you at 10:45.
Recording stopped.
Right, I'll see you around exactly at 10:45. That's around 30 minutes from now.